Hello again! It's your boy, the one and only Sir Patrick of the Department of Biology. So I hope by now you have finished watching our first lecture titled The Science of Systematics and Taxonomy. And I'd like to welcome you all, to cut this all short, for our second lecture, The History of Systematics. So I hope you enjoy, sit back and relax, and watch this video. Okay, for the second lecture titled The History of Systematics, we'll only have 3 topics for our outline. So the first one would be the history of systematics. The next part of the lecture would be on the significance of taxonomy in systematics as a branch of systematics. And the third one would be the importance of collection of specimens. Okay, for the first part of our lecture, it will be more of a historical background, a history, because some say history tends to repeat itself. And in that same manner, in that same argument, history needs to be discussed in a way to avoid it, maybe, or maybe learn from it, as I can say for myself. So that's the thing about history. If you don't study it, if the Marcos rewrite it, the people will not learn from it. The people will be blind, and history will indeed repeat itself. So I think the discussion of history for this course, Bio 110 Systematics, is actually, well, logically important or ethically important. Anyway, first of all, I would like to agree with Franz Staflou that it is indeed futile to attempt to cover in this lecture the 24 centuries, all those years of human effort to cope or wrap our heads around these multitudes of living organisms that we study. I could only try, and I hope I don't get visited by the ghost of all those scientists that did provide a considerable contribution, but was not mentioned here. Well, the first attempt at systematizing biological knowledge was by the Greeks. And now I wonder, where did the expression "you are talking" or "I feel like I'm reading Greeks" came from? Maybe it came from this, don't you think? I don't know, I'm actually not sure. The Greeks were definitely good. They were the best at the time, with the likes of Plato, Aristotle, and Theophrastus, Socrates, and those people. I would like to believe that, well, for me, systematics started with human speech. No. Scratch that. I think it started with the presence of humans' natural intuition. We all know that humans are born naturalists. Even a child has this natural instinct to distinguish a fish from a rat. Fun fact, at the age of 2, a child could start being a racist. Share ko lang. That's why I beg to differ with Franz Staflou's statement. And I think that the mere naming of individual items or objects in the natural world by our ancestors, by our indigenous people, could actually count as a way of classification, a proto-classification if you ask me. And for the case of the Philippines, it's actually important and helpful if that plant has a local name. Like, for example, the local purikit or bangbangsit plant in Nueva Vizcaya or in Ilocano regions. If you know bangbangsit, it's Lantana camara. And the purikit is the one that kinda sticks in your clothes when you ran in the grassland. Anyway, that's just childhood. Okay, still, biosystematic consists of the various attempts by the Greeks. So, the answer here is what? In this PowerPoint, as you can see, the ancient Greeks, the golden age of Greece. So, for the question 1, you can just pause the video. So, number 1 is to Entoma Malakia, reptiles, fishes, and birds. Who is this person in our number 1? What's the answer? You can pause the video. You can guess? So, have you got your answers? Yes, it's Aristotle. Starting with him, because the first question back then was the being. Where the Greeks always has these questions like, what is a being? Our existence. Do we exist? Our sentience. Are we a sentient being? The nestness of things. For example, the chairness of things. How would you, or how could you say a chair is a chair? What is it? What characteristic is it that makes a chair a chair? Its essence as a chair, something like that. So the Greek always has this problem about this whole existence, the essence of things. That's why I think it is actually important that we start with them, specifically with Aristotle, because he was the one who started the art, so I say, of classification. Entoma meaning insect, Malakia meaning cephalopods, and Malocostraca as crustaceans, as you have read in our readings. Okay? Then come this person for number 2 during the golden age of Greece. So who is this person, number 2? He is the student of Aristotle and divided the plant world according to habit and nearly wrote 500 plants use, arrange them in several groups and recognize as families in order. So who is this person? Do you have your guess? Is that your final answer? Yes, this is Theophrastus of Eresos. Back then, he uses this deductive reasoning for classification and base his scheme of classification on some important features like the presence of vascular tissues or circulatory systems of organism. It is also here that the term genus or genus and species or eidos was used to group things. So yeah, genus or genus, species or eidos. E-I-D-O-S. Eidos. Okay, for the second part of our chronological order of history of taxonomy and systematics, we have the period of Herbalists to Darwin. We have Otto Brandtfeld, Leonard Fock, which focuses on the arrangement of genera essentially in a more or less alphabetic order, as well as the likes of Conrad Gessner, where in his book, the 4-volume book Historia Animalium in 1551 to 1587, has been a really helpful way of categorizing and classifying organisms. And yes, these 3 people are actually responsible for a certain increase in scientific research and created a sense of dissociation, a mild dissociation from the ancient texts and teachings back then. Then comes the works of Hieronymus Bach, or in a more recent translated name, Hieronymus would be Jerome. So Jerome, during 1515 to 1544, with his knowledge of the floral morphology, has put some influence on the classification, where he based his method on the combinations of vegetative characteristics of those plants. However, it was only during the time of Valerius Cordius in the 1515 and 1544 where we can see the full dissociation, the full abandonment of the blind trust in the ancient botany text. He relies solely in his own observation. Well, that's expected because he is an astute observer and an independent thinker. Well, I'm really proud of this man. This person got some balls. Yes, Valerius is a renowned physician and botanist back then who wrote how drugs are to be prepared and therefore is responsible for the renaissance of botany
and he pushed botany back then to its maturity. So that's Valerius Cordus. Okay, so we'll have a quiz. The question is, who among them is a zoologist? You can pause this video. So is that your final answer? Yes, it is Konrad Gessner. Konrad Gessner was the zoologist among them. The next question would be, who arranged alphabetically the inventories of natural genera, the taxonomic groups or taxa of specific generic or even higher rank? Who among these people? Do you have your answer? And yes, they are letter A and letter B. Otto Brunfel and Leonard Fock. Now, moving on with the Renaissance and Enlightenment of Europe, with a focus on reason and logic, starting with Andrea Cesalpino in 1519 to 1603. Well, Cesalpino is a botanical systematist. Now we can really say the start of systematic is on, because Cesalpino tried to ask some logical questions back then. What he did was, upon the myriads of plant characters out there, he was thinking, there are many, super many plant characters out there. But his question was, which characters are the most important? And in that moment, the Aristotelian and Theophrastic physiology was totally in the bag. And he knew that certain parts have an utmost significance than others, specifically those involved in nutrition and two generations. These two vital aspects of plants. Here, the nutritive aspect of growth was the highlight, and thus brings about the split between the woody, the trees, and the herbaceous plants. So, yeah, the other important criterions that he mentioned in the reading was the presence of seed, embryo, and the seedling. Indeed, Cesalpino's work showcased the consistent application of his predecessors, the Aristotelian a prioristic principle in plant classification. Although Cesalpino himself is a renowned observer and an inductive naturalist himself, just like Aristotle. So first of all, you might be thinking, what is a priori and what is a posteriori? We'll first describe that or define that. When we talk about a priori or deductive reasoning, this is more like a top-down approach to reasoning. It relates to the derived reasoning from self-evident proposition. So you have propositions and you kinda derive your reasoning from those self-evident propositions. While for the case of a posteriori or inductive reasoning, this is more like a bottom-up approach, which is also a kind of reasoning, but it is more or less obsessed with observed fact. A priori is deductive, while a posteriori is inductive. And Cesalpino is more fond of the a priori Aristotelian a prioristic principle than the inductive naturalist within him. Okay, is that clear? Because sometimes philosophy has problems with science. I mean, more precisely logic. Because in logic, not all deductive reasoning, specifically with the hypothetical methodology in science, the scientific method, not all the time this method of reasoning would hold. And it would actually limit its usefulness in science. Well, it's true that the behavior of the whole could be analyzed in terms of its properties, of its parts. But properties of the parts are not intrinsic properties, but can be understood only within the context of a larger whole. Thus, using a hypothetical deductive method, this deductive reasoning, it may limit our understanding of a more complex system because we tend to reduce our observations or our observation of the world. Okay? Thus, we might end up committing the fallacy of ambiguity or existential fallacy. You will be tackling that in your philosophy class, logic class in the university too. So, I hope you do well in that course. Anyway, after our herbalist Andre Cesalpino, we come to the next person, moving on with Pierre Magnol. So Pierre Magnol, on the other hand, committed the important steps in the emancipation from the preconceived notion towards unbiased inductive research. He was the first to use the term family, yes, as the way we are using family now in this course. Okay? The family of plants, animals, and species. Okay? He was able to see that certain characters are unique to a certain family too. And unique only to that family, to that certain family. But this character may not necessarily be important or present in the other families. So it was like a defining characteristic of certain families. Like, for example, for the case of the family of Bayabas, Myrtaceae, when you smell their leaf, there is this certain distinct smell. If you smell a Bayabas tree, if you crush the leaves and smell it, it has this smell. Well, if you also crush the leaf of our test tube plant brush in Burnham too, and crush it, it will also have this distinct smell. So that's kind of like it. Or how certain plant families has white sap, yes, yung mga puting dagta, and some has dark sap. Things like that. Things that are specific to certain families of this organism. Okay? So that's Pierre Magnol. He also listed different families of plants and used conspicuous characters like fruits and stems and flowers for his scheme of classification. Okay? So that's him. Moving on with John Ray. Ooh, a more sexy name. In 1628 to 1695. He is an English philosopher and a naturalist. He used the actual term monocotyledonous or dicotyledonous, and he used different combination of characters in his categories, and was the one who attempted to use the genus as a taxonomic group of comparable importance. Okay? He was the one who first tried to use a factual instead of a relative meaning, a more factual meaning with these categories, no? His definition of the genus was more factual than relative. He kind of tried his best to stabilize the meaning of genus and species, these different categories of classification. He also used fruits and flowers for his characterizations of different plants, and he used it in forming his dichotomous table, a method of easily classifying or identifying organisms. You will learn that more, the use of dichotomous keys, dichotomous table, in your Botany 109 course here in BS Bio. So watch out for those. Okay, next would be Joseph Piton de Tournefort. So Tournefort, on the other hand, collected plants from Greece, Asia Minor, France, Spain, and authored the modern genus concept. Yes, he was the one. He also did establish different herbarium collections of specimen. So Tournefort actually acknowledged the didactical advantages of flowers in the delimitation of his categories. So this is actually, up to this day, taxonomists and systematists and bio students should learn how to use flowers to identify organisms. The only thing you need to identify a plant is flowers and fruits. If you have flowers and fruits, you will find it really easy to identify organisms. But if you only have leaves, stems, and roots, good luck to you. Okay? So that. Not included here, but the most important one, because I didn't include it, because Linnaeus was more or less a renowned part of taxonomy and systematics. When we talk about Linnaeus, we came to a more familiar part of the lecture. So Linnaeus was this naturalist, wherein he came in this world, and with him came the period with nomenclature and taxonomic system proliferation. It was at his time, and with his work, the naming, the nomenclature, and the taxonomic identification of organism really proliferated. Yes, all of those names of organism was not possible without the orderly genius of Linnaeus. And it was always Linnaeus. When it comes to naming, he will always be the basis, the reference. Kind of like the BC and AC, before Christ and after Christ. In the same way, there are these rules in naming, citing before Linnaeus and after Linnaeus, because he is an important figure, kind of like a Jesus in taxonomy. So that's Linnaeus for you. But the study of systematics will only make sense only in the origin of the community, a true phylogenetic relationship. Systematics, as I always repeat it, will only make sense in the light of evolution, these phylogenetic relationships and this process of evolution. That's why it's only during the time of George Louis Leclerc de Buffon and Michael Anderson wherein the real systematics was created. Yes, the works of de Buffon, a French biologist, and Michael Anderson marks the start of a hard case systematics and not just mere classification. De Buffon focused on the genealogy to pertain to phylogeny. He focused on questions like allied species came or arise from a common ancestor, things like that. And he also formulated the questions only in the light of evolution, the origin of the community. A true phylogenetic relationship would give a more meaning, a deeper meaning to systematics. So that's de Buffon. While Anderson, Michel Anderson, on the other hand, was considered the grandfather of numerical taxonomy. Anderson has his natural method, a prototype of the natural system in botany, where you differentiate the weight of characters without singling out any characters a priori, but actually evaluating them a posteriori after careful consideration and comparison. This process is intrinsic weighing of characters. as opposed to extrinsic, that is based on your extraneous a priori arguments as to essence and naturalness. So that's the work of Michel Anderson. He used different characters, but he put different weights on them. One character would be much more important than the other. And he used that in his classification scheme. So that's Michael Anderson. Now, this is the only part where things get exciting. With the presence of theory of Darwin and Wallace, their biological researches in the Galapagos Island and the Malay Islands change how we view systematics. In the presence of the theory of evolution, things started to change. Indeed, the 18th century progress had matured during the 19th century through evolution. It gave us a true picture of the phylogenetic reality, especially when they add the time dimension in their biological researches. Guys, don't you see the significance of this? It shows the blueprint of creations. That's how deep Darwinian impact, the concept of this evolution was really important. And this event was pivotal for systematics. So Adolf Engler considered that, yes, during these times, Adolf Engler have concluded that monocots are primitive than the dicot as a continuity of this life history, no? This tracing of the narrative of our phylogeny and our genealogy and our life history. And please, pay attention to this. The effect of Charles BC. Wherein, on the other hand, he considered seed plants as a polyphyletic clade. So when we say clade, we talk about clusters of organisms or groups of organisms, the boxes that Linnaeus did. And when Bessie took a look at this clade, he saw that seed plants, the group of seed plants, is a polyphyletic clade. And please pay attention to this. This polyphyletic, monophyletic, paraphyletic, this phyletic term, because it will be discussed later and we will spend a considerable amount of time with this concept. Okay? So pin that again in your head. So another quiz, another question. True or false? Botanical taxonomy developed earlier and faster than zoological taxonomy. Your answer in 3, 2, 1. Yes, it is true. Most of the researchers focused on the herbalist than the zoologist. So that's that. Next part is another history, but the other approaches to systematics by Zavoortink, 1990. So we have alpha, beta, and gamma taxonomy. So this is a very easy read. So alpha taxonomy focuses on the discovering, characterization, the naming of species, while beta taxonomy focuses on the natural classification of organisms. On the other hand, the last one, gamma taxonomy, is more focused on the genetic basis of characters, the natural population, the isolating mechanism, the processes, the rates of evolution, and speciation. So that's the different approaches to taxonomy. The alpha taxonomy is in alpha because it is the first part and is very important. The gamma taxonomy is far more advanced and would require different concepts of biology. Okay?
Oh, the desolation grows. Every inch revealed as my heart is pierced. Oh, my soul is now exposed. In the oceans deep, in the canyon steep, walls of gray sand. All my desperate calls echo off the walls, back and forth, then back again. To believe I walk alone, is a lie that I've been told. So let your heart hold fast, for this soon shall pass, like the high tide takes the sand.
Okay, for the next part of the lecture, we have the significance of taxonomy or its position in systematics. So again, we encountered this figure in the first lecture. It is actually a summary of the differences between a taxonomy, the field of taxonomy, and systematics. And as you can see here, taxonomy is actually a branch of systematics. Okay, so the only difference between them is that taxonomy does not deal with the evolutionary history of organism, and on systematics, it is the utmost important part of it. Just like what I said, it is only in the light of evolution that the history and the links between different organisms can make sense. Yes, well, according to Meij, indeed, I can hardly think of an evolutionary problem that has not developed out of some finding of taxonomy. Because only if you name the organism where you can start other things, you first have, just like what I said the first lecture, it is important that you first identify the organism, describe the organism, name it, before you can study it further, before you can do experiment on it, before you can serve it, before you can use it as a model organism in your experiment. And in your thesis, well, I'm thinking very far ahead, you would need to classify organism before you can study it. That's the first part. You need to have an identity of your organism before you can do anything from it, before you can extract resources from it. You have to first identify it. And that's the problem, no? That's the problem with taxonomy. There is actually a lack of taxonomic studies. Wheeler 2004 actually has this graph wherein, as you can see, oh my god, where we only have an utmost of 300 studies. That's so low, no? From 1993 to 2001. It's a very low. And let me get to this. There is actually a reason to why study phylogeny. Well, the first one is to provide a conceptual context for biology so that all facts and phenomena could be interpreted from an evolutionary historical point of view. And the second part would be so that our classification could reflect the pattern of descent with modification So that's evolution, a descent with modification that binds all species to a common history. That's the important part in systematics. That's the main difference of systematics and taxonomy, from taxonomy. No? We have to consider the pattern of descent with modification. When we say descent with modification, we're talking about evolution. Evolution is the descent with modification. And evolution is the common thing that binds all species together, that link us, humans, to the bacteria out there, and to the plants, and to every organism alive and somewhat alive, like the virus, coronavirus right now, that is killing us. Well, some of us. But yeah, evolution will tell the tale of the common history of these different organisms. So we have a suggested reading. You can read that on your free time. And now look at this. Look at this. Yes, this. We'll study this in the next chapter of our life during Sir Patrick time, my time. And if you look at this, these are ACT, CAG, these are nucleotide sequences of organisms. So the nitrogenous bases of the DNA of organisms. That's thymine, adenine, guanine, cytosine. Okay? And if you look at this table, if you look at this sequence data of organism, does this make sense to you? Does any of this make even a small sense to you? Does this give you anything? Because when data like this, sequences of organism like this, is viewed in isolation, it wouldn't make sense. It wouldn't make even the slightest sense. It would only make it interesting in a trivial sense, no? And only in the light of comparative morphology, ethology, unless we pour some effort in studying comparative morphology, ethology, ecology, and all of those, we can explain and make sense of the data. No? Only then we can explain the phylogeny of organism, the connections of this organism. And that's the problem with systematics. The problem with systematics is that we are having more and more systematists who can't identify organisms, who can't do the old way of comparative morphology, comparing organisms, who can't do ecology. That's the sad thing about here. So yeah, taxonomy. When we hear taxonomy, people usually say useless knowledge. I can search that in the internet. It's just naming of organisms. It's just for collections of museum collections of different organisms. It's a very old way of thinking. It's too difficult. Taxonomy can't compete against other hypothesis-driven science like genetics or physics or other fields of biology for that matter. Well, all of this statement present a very negative attitude and branding on taxonomy. It is difficult. The thing here, when we do this, taxonomy tend to get a bad rap. Where in fact, taxonomy is very important. And taxonomy is actually a scientific, is a hypothesis-based science. It's not just a simple naming scheme and boxing of things. Taxonomy is a hypothesis-driven science. For example, how these two structure right here. So this is a bat wings. This is a bird wing. And this is, I think this is another bird wing, but a very long one. So different organisms across different families has certain recurring features. As you can see, green, blue, and red. These are what we call same or homologous structures that is present among different taxa or different organisms. So in that manner, taxonomy gets to be a hypothesis-driven field. Yes. And if you think about it, every rank in the classification scheme of Linnaeus is a hypothesis in itself. It's a hypothesis worth testing. And that's the exciting part about Linnaean taxonomy and of taxonomy. We can test if the classification that Linnaeus used was good or really bad. It's a hypothesis, all of those classification schemes. And when we describe new species, and describe new specimens, we need to examine all the variation of all the related species for this organism. For the case of this Rafflesia that is in our midst, in our forest here in the Philippines. And that's the important thing here. And I had a experience with this where we went on, I went on with an expedition of Sir Maverick Tamayo. We came to this mountain in Bocod, Upper Agno River Basin, and we encountered this very minute species of orchid that is a commensal to trees. And he noticed that this species is actually a different organism. And I was actually fortunate enough to be a part of that discovery. And he just published this species of organism, this orchid species, no? And it's a very colorful orchid. And we describe and picture every nooks and crannies of the flower and draw it just to publish. So that's that, no? It's actually also an exciting part of taxonomy where you can name your organism, your discovery, I mean. Imagine that, you can name a plant for your crush or your loved one. Think about it. Okay, moving on for the last part of our lecture, the importance of collection in systematics. The first part is the critical role of collection in identification. Well, this figure shows the complete determination history of all names of all species of Aframomum, or the ginger family. Okay? So before the current monographs, more than 58% of the species, more than 58% of the specimens were misidentified or only identified up to the genus or family level. So it is indeterminate. No? It's indeterminate. The identification was not complete. Or, in short, it was wrongly named. And yes, for some of my friend taxonomists, like Sir Mav, when we are in the field, when I notice there is a ginger family and I would like him to identify it, he would say, no, I don't like that. Some taxonomists do fear some families of plants because some of them are very problematic and are difficult to group or to study because they are loose or a very loose clade or very problematic clade where you will need to fix. Yeah, a lot of work would be put into. So this figure perfectly demonstrates the first one, that before taxonomic revision, a large number of species are, look at this, yeah, look at that. A large number of species before the revision of taxonomy has been wrongly named. And number two, that from 1999 up to 2000, 1969 to 2000, look at that, the number of species that was described doubled. The next part for the critical role in identification is for agriculture and environmental biology. For example, this plant pathogens and pests. This pygidium of this insect, Aspidiotus rigidus. Yes, this insect, Aspidiotus rigidus. And so this left one is for the Apidiotus rigidus, and the right one is for Apidiotus destructor. So this is actually important, no? Because this fly, this insect, has a devastating effect on the coconut tree here in the Philippines, wherein the infestation of this armored scale insect is now spreading in the southern Luzon from Batangas now creeping into the Visayan Island. And the sad thing here is that it caused damage to our livelihood, especially from the province I'm in Bicol. When the plant is infected, the leaves stops undergoing photosynthesis, and therefore it cannot make food, and the leaves become yellow and dry, and therefore the coconut meat wouldn't be much of a profit, and the coconut water, the buko juice, would taste sour. That's the problem, no? And when you have taxonomy, you can study them and differentiate which is much more susceptible, how to manage them. And the last part for critical role in identification is, well, if you have a great collection of species like this one here, you can have a vivid picture of life on Earth, okay? And the second importance of collection would be the revisions of taxonomy would require all of these collections. So when, for example, an organism is extinct, you can possibly collect more because, well, the species was gone. So you need to refer back to the collections of the people back then. And phylogenetic reconstruction using data from that collections, we can also extract DNA from those organisms that was preserved and collected, and we can now create a more or less reconstruct the organism. So that's the importance of collection. Like, for example, the case of this Epipodium ketingiensis and this Epipodium sp. So Epipodium ketingiensis, Epipodium sp. So as you can see, so Epipodium is a ghost orchid. It's a terrestrial leafless orchid. It's actually very interesting because it's a mycoheterotrophic orchid, meaning it has a symbiotic relationship with the fungi, and thus it lacks chlorophyll. So it's famous for its very unpredictable appearance. And we have plenty of this in the Philippines. And as you can see, there's actually there are differences between these two species, no? These different species. And if we collect samples of this orchid and compare it to each other, we can create different revisions of the taxa in the near future. Okay? So the last part of our lecture, the most important one is for critical data, especially for historic occurrences, conservation biology, and biogeography. So different herbarium data and different collections can provide an ecological baseline data, especially in poorly sampled region. And also, for the cases of endangered species like Papilio chicae, this very beautiful Luzon peacock swallowtail. So it's so beautiful. There are also those ones in Mindanao that could be different species, which must be undescribed by now because, well, we don't do much taxonomic studies. But do you know, in Thailand, in the Karen Yungtai community, it's an indigenous community, they have a specific rules and laws to protect butterfly. Yes, just those butterfly for tourism. Why don't we do that, right? And the last part of that importance is for those specimens which has symbiotes in them. Look at this, yep. And those specimens with spores and fungi, fungal spores, pollen and spores, and parasite. And when we have collections of these organisms, we can create or harvest those symbionts and pollen if we can't find them again in the nature. And yeah, whew, wow, that's it. That's it for our lecture for today. So as a summary, we have the history of systematics, we have the significance of taxonomy in systematics, and the importance of selection. So that's it for this second lecture titled The History of Taxonomy. I hope you enjoyed it. And if you have questions, comments, and suggestions, please leave it down in the comment section below. And if you like the video, please leave a thumbs up. And yes, see you again. Till next time, peace out.